Thank you so much, and thank you to the organizers, and thank you to Jill Sackler, and thank you so much to Allison for the invitation. I'm really honored to be here. I guess my talk's going to be a little bit different from the talks that preceded, just based on my background, but I suspect you'll see a fair number of common themes and threads being mentioned. My background's actually in clinical medicine. I'm an infectious disease doctor, and sort of strayed from the path and became an epidemiologist. And I've had a long-standing interest in the influence of environmental factors in the genesis of infectious diseases, and mostly by way of convenience, because I've worked in health departments and collaborated with health departments in high-income areas. I've become interested in environmental impacts on infectious diseases in high-income areas like Toronto, Ontario, and Philadelphia, Pennsylvania, and so forth. And again, that's largely been a function of convenience rather than importance or scientific curiosity. It's just the data were at hand. So that'll be the perspective I'll be speaking from. There's a long and rich history in the medical, I'm not sure if you'd call Hippocrates sciences, in the medical field, of trying to link environmental influence to occurrence of disease. And in the Hippocratic corpus, which is thought to have been written around 400 BC, thereabouts, the interactions between environment and health are repeatedly stressed. And there's a treatise called Airs, Waters, and Places, which is sort of a field guide for Greeks taking to their ships and planning to colonize new cities. Hippocrates had all kinds of advice on what constituted healthy environments and what he thought the drivers were of seasonality of disease. And this idea had remarkable staying power. I mostly like this because it sort of sounds a little bit edgy. This is a master clinician from, I believe William Cullen was a professor either in Glasgow or Edinburgh. He was one of the great Scottish clinicians of the 18th century, talking about the bastard peripneumonie, which is presumably what we'd call bacterial pneumonia, that it's a disease that appears at the same seasons that other pneumonic and catarrhal affections commonly do, that is in autumn and in spring, he says. It's occasioned by sudden changes in weather from heat to cold. So you sort of see this long history of people trying to tie environment to changes in environment to observed surges in clinical volume of one sort of case or another. And more or less regarding these surges in incidence and the changes in environment that seem to precede them as kind of a fixed, predictable system. But of course, human populations change in interesting ways, and sometimes those changes are associated with changes in disease patterns. This is a really neat paper by a guy named W. Evans, who apparently was the head of the American Public Health Association way back when, lived in Chicago. And he noticed, and this is true in my city too, in Toronto, he noticed that the expected surge in summertime mortality, which was mostly infectious diseases, seemed to flatten out and then invert in the 1910s. This is leading up to the great pandemic of influenza. But he's not adjusting for population in Chicago here, and Chicago is obviously growing rapidly in the late 19th century. But you see this very nice stereotyped wave kind of flip upside down, and Evans sort of scratched his head and wondered what that was because of. I don't know if anyone in the audience has any thoughts on that, why you would see a transition from people dying in the summertime of febrile illness to people dying in the winter of febrile illness. Maybe I'll leave that with you and let you think about that. It happened in a lot of different places around that time. But when we try to make these inferences, I guess my talk is mostly going to be about how difficult I find it to, possibly because of my lack of formal ecological training, so I feel like a bit of an interloper. But I think these are also very hard problems. And I'd like to talk about some real challenges to understanding the links between the physical environment and disease incidence. And I've grouped these, although these aren't mutually exclusive groupings, I've grouped these under the headings of scale. I'll mostly talk about temporal scale because, again, I'm not a geographer, but you could say similar things about spatial scale. Some of the perils of aggregation, some of the solutions we've tried to use that relate to case-only epidemiologic designs. Some of the challenges that relate to the complexity of biology, including the cumulative effects and multiple lags that environmental shocks can have on downstream occurrence of disease, and how we might approach that with models. I'll try to talk specifically about complexity, again, an area in which I'm no expert, but some of the challenges that relate to oversimplification of systems and models, and some of the statistical challenges that relate to trying to build very realistic statistical models and infer cause and effect relationships, the statistical cost of complexity. Lastly, again, I'm not a geographer, but I'll put a toe in the water on geography. Some of the challenges that we've run into related to arbitrary boundaries. I'm working mostly with mandatory sort of notifiable disease data, which tends to come from geographic entities that often have arbitrary bounds to them. Mobility of populations, heterogeneity of exposures, and possibly heterogeneity of effects of the same exposures, which I think is actually potentially quite interesting. Potentially the most interesting thing I'm going to talk about, though, is the thing I know least about. Lastly, as a brother of an economist, I learn a lot from my personal economist, and I think we can all learn a lot from economists. Economists sort of sniff around and look for natural experiments. Randomized trials are now all the rage in economics, but for a long time economists had to really keep an eye out for natural shocks, natural experiments. All these will overlap. I'm going to go very quickly through this slide because this is going to be intuitive to all of you, I think. Some students of mine made this a few years ago, Laura Kinlan and Alex White. Alex and Laura were looking at pneumococcus and campylobacter. I can't remember which was which, but both of those are bacterial infections that are profoundly seasonal. And Alex made the point that if we simply, regressed the number of cases per week on sunshine or temperature or various air pollutants that are highly seasonal or ice cream sales, we could get very impressive estimates of effect and, perhaps attribute campylobacteriosis to ice cream sales. And I can't remember if there have been Campylobacter ice cream outbreaks. There probably have been. But of course, we don't do that because when we have two processes that are oscillating in phase and we simply throw them up on a scattergram, we're going to see a very nice correlation between the environmental exposure and disease incidence. So we try to detrend these processes and fit models that incorporate seasonal smoothers, long-term time trends, and so forth, approaches that are probably familiar to most in the room. And when we do that, we find some interesting relationships. So sure enough, even after we detrended, these are Philadelphia Campylobacter data, even after we detrend them, what we find is that, yes, increasing air temperature and humidity seem to be reasonable predictors of campylobacteriosis, but then we have backwards effects that go along with the temperature of the Delaware River at 0 to 4 week lags. An unexpected finding, and then we sort of dug into the microbiology and found out that Campylobacter in surface waters actually survives better and survives longer at lower temperatures. So perhaps plausible. But the importance of trying to detrend. Now, of course, all of this winds up still using disease counts that are aggregated over some unit of time. So usually cases per week, cases per month. And that can be problematic because the interesting environmental effect may occur on one day or part of one day and result in a downstream surge in cases on another day after some lag. But if you aggregate, you're effectively smoothing out both the exposures and the responses, and you're going to lose the ability to identify those effects. There are some study designs that are well suited to the study of available public health data And particularly, this is what I'm going to mention more, because case crossover design, which is an analog of a case control study, which many of you may be familiar with, is an epidemiologic method that's used to study cause and effect relationships when outcomes are rare. Now, as it happens, a lot of the things we are interested in public health turn out to be rare events. So this analog of a case control study allows us to study rare events, but also allows us, in terms of exposures, to study brief, transient, repeated exposures and their impact on rare events. So what we'll do is we will look upstream temporally from the time a case occurs, and then we'll choose a control time period, or 2 or 3 or several, that are close enough in time that we've effectively detrended relationships, same season, same city, actually same person, the case at a time when the case did not become a case, and look at differences in upstream environmental conditions prior to the case becoming a case versus prior to the control staying a control. But again, the case and the control are the same person. So we implicitly also control for any factors within an individual that stay constant over time, like ethnicity, age, other modifiers of risk. We tend to use random directionality of control selection, because if we use stereotyped backwards directionality of control selection when we have temporal trends, we can actually that can result in biased inferences. But here we have a 3-week time stratum where the case occurred on Friday. Here are 2 other Fridays that are nearby in time. Those are going to be our control days. And we're going to look upstream in terms of what the environmental exposure looked like just upstream from the case versus upstream from the control periods. And we can then estimate odds ratios using conditional logistic regression, as we would use for highly stratified data. This is a really nice project that was done using this method by Jonathan Soverow when he was I believe a medical resident at Harvard. Jonathan managed to get the CDC to give him individual level case data on West Nile virus in a few regions of the United States, and he applied. That's sort of what his time series for West Nile cases look like. This is when West Nile was first emerging in the U.S. And Jonathan was able to look upstream and see that things like temperature, and one of these that's temperature, and humidity upstream seem to have interesting effects on downstream West Nile risk. This is something that Stefan Kuster, who was a student with me, did a few years ago, using exactly the same approach to look at invasive pneumococcal disease. We have a very rich data set in Toronto on people. William Osler famously called pneumococcus the captain of the men of death, which I think is from Pilgrim's Progress. It's the captain of the men of death. This is such a nasty infection, which thankfully there's better and better vaccines against. But it is something that continues to kill people, often people with comorbidities. And Stefan looked upstream to case occurrence. Case occurrence would be here at time 0. He looked at different lagged exposures. Here the exposure is influenza. We were trying to look at whether or not influenza actually increases your risk of downstream invasive bacterial infection. And Stefan found that it did, in Toronto anyway. But what he also found, which is kind of neat, is if you go back a further lag to 3 weeks, you actually see a protective effect, which is probably a manifestation of harvesting. So the people who were predisposed to die of invasive pneumococcal disease based on an earlier surge of influenza are already dead. So you now have, or not necessarily dead, but have already gotten their invasive pneumococcal disease. So you see this harvesting effect where a longer lag makes it look like influenza is actually protective. So those are some of the sorts of things that you can do with case crossover study design. Now that's just looking at single lags, but of course we may have very complicated lag functions, and we may have a single exposure, for example, a dramatic rainfall. We live in this strange era where 100-year rainfalls now seem to happen every other year, which is, we probably need to change our syntax. Where a single exposure can have multiple and even contradictory effects over multiple lags. Mentioned harvesting. With vector biology, we have all different life stages. You know, you can have an exposure like temperature have an impact upstream on maturation of larvae that turn into pesky mosquitoes, and then downstream there's this sort of interesting J-shaped relationship between temperature and biting behavior, where when it gets hotter and hotter, mosquitoes bite faster and faster. But then my understanding from data in Bombay is that eventually they say, "Woo, I'm schwitzing. I don't want to bite anymore," and they slow down their biting. So we can have interesting, not necessarily linear effects, but acting on multiple time scales. So something that we've recently got into, which is maybe not news to some of those in the audience, is the use of distributed lag models, where we actually model an exposure not as a one-dimensional effect, but as a two-dimensional exposure surface, where we have exposure times lag. And that is then the exposure that's incorporated into a regression model. And our output from that becomes a three-dimensional risk surface, where we have on the x and y axis exposure and lag, and on the z axis we have relative risk. And I'll show you pretty pictures of that in a moment. This is just looking at, this is an old slide thinking about what the potential effects of climate change could be on a vector-borne disease, like, say, West Nile virus infection in Canada, where what we have now is we have a longer and longer and longer period between spring melt and autumn freeze up, which means that eggs get laid earlier, larvae develop faster. There's a lot longer period for amplification to occur when this particular virus goes bird to mosquito, bird to mosquito, bird to mosquito, and at some point you have so many infected mosquitoes that you have a reasonable percentage of cross-biting mosquitoes start to bite humans. And under that sort of model, you might expect to see West Nile season get shifted earlier and earlier in the year, from August, say, back to July, back to June, which would result in more infections. So we could imagine seeing different effects, a given exposure, which is a surge in temperature, having effects on eggs laid, but also on biting rates of current mosquitoes, and that those would manifest in disease risk at different time lags, surges in disease risk at different time lags, here and now, and also later, because there are more baby mosquitoes maturing, which are going to be the future biters. This is just to show you some work in progress that we've been doing using the National Hospital Discharge Survey, which, remember, these are all hospitalized individuals, so this is not necessarily ideal, because most people with infectious diseases do not get hospitalized for those infectious diseases, but it's a nice data set and it's free. We have a national probability sample of hospitalizations in the US with survey weights that lets us estimate incidence of different diseases. There are about 40 million observations over a 40-year period in this data set. And there's not a lot of granular data on individuals or hospitals, but the data are broken down by US Census regions. So our idea was to, since El Nino is a periodic shock, trying to think like an economist, the idea was to try to look at El Nino effects, which may approximate in some ways what we can expect in future more frequently with climate change. That is, more extreme rainfall events, higher temperatures, and so forth, and look and see, particularly in the western region, which is thought to be more teleconnected to El Nino, whether we see surges in disease in the western region under El Nino conditions that are different from surges in disease elsewhere. So our controls are the non-western regions, and our experimental group is the western region. And what we see here, this is looking at vector-borne disease, which is, I've lumped together, can't remember which of these is rickettsial disease and which of these is arthropod-borne viruses, but I've lumped them all together to get enough numbers. And we can look at a risk surface for the western region using a distributed lag model and see that downstream we've got, initially at this 10 to 12 month lag, there's this surge in risk associated with more El Nino-like conditions that disappears downstream. And here it is just as a contour plot. You can see the surge in risk upstream. That's what it looks like in cross-section. So this is something called multivariate El Nino index, which is sort of a multi-component way of deciding whether or not things are El Nino-ish or not, and it involves C and land and sea surface temperature, pressure measurements, and so forth. So we see this nice surge. We can then integrate, well, the computer can then integrate the area under this curve and give us a summary relative risk. So we could say, well, an MEI value of 3 in a given month in the Western Health region, there's a downstream relative risk of vector-borne disease in the Western Health region that's about 3, with obviously wide confidence bounds. Okay. So this is, I mean, this looks fancy. My dad was really impressed by this graph, and it looks fancy, but I wouldn't call this complex. We're regarding multivariable El Nino index, the relationship between that and disease. In the model, it's specified to be linear, okay? So this is not complex. We have this very simplistic idea that as it gets more El Nino-ish, risk goes up. A model, please test that. We obviously have a cubic function in terms of the lag function, but risk is linear. And as things get more complex, which is to say get more realistic, all of this gets harder and harder. In looking at biological relationships, there are numerous interactions and relationships that may be unexpected, may be unknown. We may misspecify both mathematical and statistical models, oversimplify things and miss important complexity. But what I've noticed with the distributed lag models is when we try to incorporate more realistic effects, we simply lose out in terms of the statistical power to identify effects. It's also very difficult to capture non-independence of environmental exposure measures. So in some regions, we may see weather patterns that are hot and dry, and in others, they're hot and wet. So should we actually expect the effective temperature in a hot and dry region to be the same as hot and wet? When it's very warm in Philadelphia, should the effective temperature be the same as when it's very warm in Arizona? I don't know, but those are the sorts of complexity we're confronted with. Back to Campylobacter, in terms of how poorly we understand some of these systems. As a medical student, this is what I was taught about Campylobacter, is that Campylobacter is summertime seasonal, and it's what I tell students is the Homer Simpson effect. That is, the usual heuristic is that the gentlemen, because we'll talk about sex predominance in males, wheel their barbecues out onto the back porch and start undercooking and consuming chicken. That's what I learned as a medical student. It's probably more complicated than that. As mentioned, there is this very interesting male-female predominance. So men, I did this as a quick and dirty meta-analysis for the talk, so it's not by any means a systematic review. It was an easily obtainable paper review for a forest plot. But whether you go to Ireland, Israel, a couple of Canadian data sources, the US, it's actually remarkable how homogeneous the effects are. This is some data from China. You have about a 15% excess risk in males over females for Campylobacter. Now, given that this is supposed to be an environmentally abundant pathogen, that's really weird. If we're all, if Homer is cooking the chicken and undercooking it, he's presumably going and feeding it to his family. So we shouldn't see a 15% excess male to female risk. But maybe there's other stuff going on. These are small numbers, but this is from the Canadian CenterNet Sentinel surveillance study. We see this funny surge in Campylobacter numbers in 20 to 24-year-olds, 20 to 24-year-old males versus females. What on earth could that be about? Well, with gonorrhea, with syphilis, we use male to female ratios as indices of to what extent an epidemic is driven by men who have sex with men versus heterosexual transmission. Could sexual transmission of Campylobacter explain this excess risk? It probably doesn't explain the excess risk that's often seen in male infants, though. So is this biology? Is this immunology? This is This is from the UK. Excess risk in 10 to 20-year-olds. Is that sex? Is that how boys play? I don't know. Another interesting model comes from a fellow named Gordon Nichols at the HPA. Gordon Nichols has pointed out that Campylobacter could well be a vector-borne disease. This is complexity that we don't usually capture. He's pointed out that, that in the UK, these are multiple jurisdictions along the y-axis. Everybody's Campylobacter outbreaks start at the same time. And Campylobacter season starts at the same time everywhere. And he's pointed out that that's highly correlated with the profound drop in fly generation times that happens when the weather gets warmer. But if we try to start getting fancy with models, at least statistical models, this is what happens. This is some enteric illness work that we've been doing with the National Hospital Discharge Survey. Again, one of these cross-sections for El Nino-like weather. Those of you in California may be interested to know El Nino seems to be associated with a reduced risk of enteric illness in the western region of California, which may be an interesting thing to talk about later. But I would have expected to see a J-shaped relationship between precipitation, for example, and enteric risk. When things are very dry, enteric risk goes up. When things are very wet, enteric risk goes up. But if I try to model this as a quadratic risk function, this is what happens to my confidence bounds. So I can't really say anything. As I introduce complexity, I lose the ability to actually observe relationships. Just as a follow-on, could there actually be regional heterogeneity in effects of identical exposures? These are the same data as I'm presenting here. This is the western region effect in terms of relative risk of an upstream El Nino event in California. Risk seems to go down. If we look at all health regions except the West, risk actually goes up and has confidence limits that don't overlap. 1. So could the same heavy rainfalls, I'll ask you to press play on the movie, even though we regard El Nino as very much a Western, the squeeze wasn't really worth the juice there, was it? But even though we regard El Nino as a phenomenon that may principally affect the West, you can see that rainfall anomalies actually occur across continental North America. So we may have surges in rainfall in these other regions. So perhaps expecting these to be control regions where nothing happens. This is the Northeast, for example. Perhaps that's not right. Lastly, I want to speak a little bit about some of the challenges associated with geography. There are issues related to where people live versus where the boundaries are drawn. This is a big issue in Canada. Cases are mobile. We think that we assign them an exposure that happened in the jurisdiction where they're reported. That may be wrong. There's a correlation between risk and reporting, and I'd like to show you a little bit of that for those of us who rely on public health data sources. And risk tends to correlate with wealth as well. So the people we may be most interested in may be the people we're most likely to miss, even in high-income areas. We may have heterogeneity of effects by geography, as I was attempting to show there. And lastly, I'd like to say, let's leverage geography and think like economists. This is where I live, in this little red dot here. This is Ontario. It's a slice of territory that's about 2 times the size of France. When we look at Ontario, we tend to limit, if we do things in terms of environment and health, we do central Ontario. And we can get away with that because most of the action is down here. In 2000, there's a population of 8.1 million living in this very small area right around Toronto. So if we start then trying to average out environmental exposures from this whole jurisdiction, use all the data, well, up here we have very sparsely populated areas. It probably doesn't make a lot of sense. So we have to think about our geography. This is an old data set from basically an editorial I wrote at one point about rabies. There's, Megan will know this, there's this wonderful online data source, RabNet, I gather, that tells you how many rabid dogs a given country has reported. And you can fit this is sort of a quadratic Poisson regression, and you get this sort of nice line that says, oh yes, as log per capita GDP goes down and down and down, you get more and more and more rabies cases, and that's probably not a huge surprise. But what about these countries? They're missing. Well, if if we do a logistic regression analysis looking at the probability of no reporting at all versus per capita GDP, I'm not showing you the raw data here, but it's actually a very well-fitting model. What we see is that the probability of not reporting at all rises very sharply as per capita GDP goes up. You know, countries that have no resources to even tell you what's going on. Perhaps in the, well, perhaps I will tell you about this. We can have other kinds of confounding of environmental, that's non-reporting by wealth. We can have confounding by wealth. This was something we published a couple of years ago on the seeming relationship between proximity to the equator and fraction of bacteremias that are gram-negative. So they're gram-positive bacteria and gram-negative bacteria. This is an early branch point when microbiologists are identifying bacteria in the laboratory. And what we see is we get a sort of a beautiful quadratic relationship between here's the equator, and the further north we go, the more the gram-positives predominate and the more the gram-negatives go down. The further south we go, the more the gram-positives go and the more go up and the more the gram-negatives go down. But do I have the model? No, I don't have the model. But what's actually in this model is not simply latitude. What's in this model is latitude and percent of GDP spent on health, which is also a function of GDP. And so this possibly, although we had started this project thinking we had all kinds of wonderful explanations for why people could get more gram-negative bacteremia closer to the equator, something that seems to be very important is the likelihood that people are antibiotic exposed as well, which kills the gram-negatives, so you wind up with gram-positives. Could environmental effects be heterogeneous? This may be important. Jonathan Dushoff, a number of years ago on a paper that included several in this audience, wrote a paper noting that you could explain influenza seasonality, for example, very nicely as dynamical resonance. And for those who aren't conversant with resonance, you probably still in high school physics saw the film strip of the Tacoma Narrows Bridge falling down, the man sort of staggering along the bridge that's swaying. When you have a structure that has an intrinsic oscillatory frequency and it gets tapped at that intrinsic oscillatory frequency, even very lightly, it can oscillate wildly. And what Jonathan and his collaborators pointed out is that very small seasonal forcers, based on the virological characteristics of influenza, that's a disease that's sort of, based on our best bet, has an intrinsic oscillatory frequency of about 1 year, or period of about 1 year. So given the number of environmental exposures that have annual periodicity, it's highly likely that you would see this being a seasonal disease because it's going to get tapped at a frequency of about once per year. And one wonders, and I'd like to throw this out to the audience, given the multitude of seasonally oscillatory environmental effects, could successful, widespread, ubiquitous pathogens like flu be the pathogens that are able to be forced by different environmental exposures in different geographies? It doesn't make a lot of sense for influenza to be forced by cold weather in Arizona if there is no cold weather, right? It doesn't make sense for influenza to be forced by bright sunshine or the absence of bright sunshine in London, with apologies to the English folks in the crowd. It's you know, in a place that's foggy all the time, you wouldn't expect flu to latch on to UV radiation as the forcer. At any rate, that's Jonathan's picture. And I've wondered about this. This is a paper, again, that, the next speaker is an author on this paper as well. This is by Jeff Shaman and colleagues, looking at deviations from typical absolute humidity values as a predictor of flu season onset, and finding that that seems to be a very good predictor in the northeastern United States. In Alberta, where it's dry all the time, this is a paper by David Earn and Jonathan Dushoff and others, looking at the onset of the autumn pandemic wave in 2009. Their model actually didn't fit very well when they used absolute humidity. They needed to use surge in temperature to make or decrease in temperature to make the the model fit. So, perhaps in Alberta, which is always dry, it doesn't make sense for a pathogen to latch on to humidity as its driver. Finally, I'd like to show you some old work, but that is sort of being revived. Laura Kinlan had done this using case crossover methodology to look at UV radiation, where bright sunny days seem to be associated in case crossover with a reduction in risk of invasive meningococcal disease. Whether or not that's true, you can see there are about 1 million comparisons there, so it's probably not fair. But when Laura got a hold of other data sets in other cities and tried to look for the same effect, she didn't find it. She found other effects, other periodic exposures that were associated with increased or decreased meningococcal risk, but she didn't find that. And in fact, when she looked in Philadelphia, Toronto, London, and Sydney, she found different exposures being associated with case risk in all these different places. Lastly, I'd like to mention natural experiments. El Nino perhaps is an example of a periodic shock that occurs irregularly that we can leverage. There are also natural experiments that happen spatially. Border cities are a great example. This is wonderful work by someone I don't know from the CDC, J.M. Brunkard, looking at dengue in Brownsville and Matamoros, Mexico, which is basically one city that stands astride, I guess, the Rio Grande with a border fence, but mosquitoes don't tend to care much about border fences. And what they were able to do, now they've controlled for geography, they've controlled for pathogen abundance, because the mosquitoes are there, they're everywhere. Now they can start to dig down and finding a large difference in seroprevalence between folks who live on one side of this border fence versus the other side of this border fence. They're able to look and see, well, what are the characteristics of infected individuals? They earn less. Their streets aren't drained. They store water on their roof rather than getting water through pipes. A lot of these things are going to relate to mosquito habitat, but you have this nice sort of basically experiment and control. I don't know what you want to call the experiment, what you want to call the control, that implicitly controls for vector abundance. So I'd like to acknowledge my many friends and collaborators in the Canadian Institutes for Health Research. And this is a cartoon that says, that's a very good question, which is why we're going to move on to the next question, which has always been my approach.